Bismillah ar-Rahman ar-Rahim. The topic of our discussion today is major histocompatibility complex, MHC. MHC molecule, other molecules and other proteins involved in antigen processing and presentation are encoded in a cluster of closely linked genes, which in humans is located on chromosome 6. This region is called the major histocompatibility complex, MHC, because it causes T cells to reject tissues transplanted from unrelated donors to recipients. For some MHC class 1 and class 2 molecules, numerous genetic variants are present in the human population. Each variant functions differently. In the peptides it binds, and the T cells it triggers. Differences that individually and collectively have helped the human species survive predation by diverse and numerous pathogens. Although the magnitude of MHC diversity is much smaller than that of immunoglobulins or T cell receptors, it has a major impact on the immune response, disease susceptibility, and the practice of medicine. In this discussion, we try to cover those subjects that I mentioned, special proteins by the name of major histocompatibility proteins. Actually, they do the presenting and presentation of the antigen to T cells, and T cells receptors to view the presented antigen. Histo means tissue, also involved in the rejection of transplanted organs, and matched organs means MHC molecules of the donor and the recipient. There are two types of MHC molecules, as I said, class 1 and class 2. Class 1 MHC exists in varying amounts on the surface of most cells. I will show you a graph and a table about this. Class 1 MHC works as a billboard for killer T cells to understand what's going on inside the cells, the presenting cells. When a cell infected by a virus, peptides loaded onto class 1 MHC molecules and transported to the surface of the infected cell. Killer T cell can use their receptors, as it was mentioned in previous slides, to look into the cell to discover that it has been infected and that it should be destroyed. Class 2 MHC molecules similarly function as billboard like MHC class 1, but this time for helper T cell. Only certain cells in the body make class 2 MHC molecules, antigen presenting cells, APCs. Macrophages, for example, are excellent antigen presenting cells. Helper T cells can scan the macrophages class 2 MHC billboards for news of the bacterial infection. Class 1 MHC molecules alert killer T cells when something isn't right inside the cell, and class 2 MHC molecules displayed on APCs inform helper T cells that problems exist outside of cells. Although a class 1 MHC molecule is made up of one long chain, a heavy chain, plus a short chain, beta-2 microglobulin, it's the name, and a class 2 MHC molecule has two long chains, alpha and beta. You will notice that these molecules are rather similar in appearance but they are different in details. The T-cell receptor is a membrane-bound glycoprotein that closely resembles a single antigen-binding arm of an immunoglobulin molecules. It is composed of two different polypeptide chains and has one antigen-binding site. T-cell receptors, as you see in the picture, are always membrane-bound, and there is no secreted form as there is for immunoglobulins. Like immunoglobulins, each chain has a variable region, you see in this slide, which binds antigen, and a constant region. During T-cell development, gene rearrangement produces sequence variability in the variable regions of the T-cell receptors by the same mechanisms that are used by B cells to produce the variable regions of immunoglobulins. However, after the T cell is stimulated with antigen, there is no further mutation in the antigen binding site, and there is no switching of constant region isotype as occurs for immunoglobulins. These differences correlate with the fact that T cell receptors are used only as receptors to recognize antigen, whereas immunoglobulin serve as both recognition and effector molecules. In this slide, you see the comparison of the T-cell receptors and IgG antibody molecules. The T-cell receptors is a heterodimer composed of an alpha chain of 40 to 50 kilodalton and a beta chain of 35 to 46 kilodalton in size. The extracellular portion of each chain consists of two immunoglobulin-like domains. The domain nearest to the membrane is a C region, and the domain farthest from the membrane is a V region. Both the alpha and beta chains span the cell membrane and have very short cytoplasmic tails. The three-dimensional structure formed by the four immunoglobulin-like domains of the T-cell receptors resembles that of the antigen-binding Fab fragment of antibody. You will see in this picture more details of T-cell receptors, variable region, constant region of alpha and beta chains, and then stuck segment and transmembrane region, cytoplasmic tail, disulfide bond, and those details. There are two classes of MHC molecule having similar three-dimensional structure. This slide shows class 1, MHC class 1 structure. MHC class 1 and class 2 molecules are membrane glycoproteins whose function is to bind peptide antigens and present them to T cells. Underlying this common function is a similar three-dimensional structure which is formed in different ways in the two types of molecules. The MHC class 1 molecule is made up of a transmembrane heavy chain, you see that, or alpha chain, which is non-covalently complex with the small protein beta-2 microglobulin. The heavy chain has 3 extracellular domains, alpha 1, alpha 2, and alpha 3. The peptide binding site is formed by the folding of alpha 1 and alpha 2. The domains farthest from the membrane and is supported by the alpha 3 domain and the beta 2 microglobulin. The MHC class 1 heavy chain is encoded by a gene in the MHC, whereas beta 2 microglobulin is not. In the right upper panel, you will see the ribbon model of the structure. Left upper one is a spatial graph showing the structure of MHC class 1, and in the lower left you will see the place of the peptide binding cleft with 2 alpha helices in both sides and beta sheet at the bottom. In this slide, the structure of MHC class 2 is shown. In contrast to MHC class 1, the MHC class 2 molecule consists of 2 transmembrane chains, alpha and beta, each of which contributes 1 domain to the peptide binding site and 1 immunoglobulin-like supporting domain. Both of these chains are encoded by genes in the MHC. Thus, both classes of MHC molecules have similar 3-dimensional structures, as I mentioned before, consisting of 2 pairs of extracellular domains, with the paired domain farthest from the membrane resembling each other and forming the peptide binding site, as you see. In both types of MHC molecule, the domains supporting the peptide binding domains are immunoglobulin-like domains, alpha 3 and beta 2 microglobulin in MHC class 1 molecules in previous slide, and alpha 2 and beta 2 in MHC class 2 molecules in this slide. The immunoglobulin-like domains of the MHC class 1 and class 2 are not just a support for the peptide binding site, they also provide binding sites for the CD4 and CD8 co-receptors. I will show you a result about and a slide about that. Thus, the sites on the MHC molecule that interact with the T-cell receptor and the co-receptor are separated, allowing the simultaneous engagement of both T-cell receptor and co-receptor by an MHC molecule figure, in this figure. So the structure of MHC class 1 and MHC class 2 molecules are shown in these 2 slides that I discussed with you. And again, in the right upper panel, you will see the ribbon model of class 2. Next is spatial panel, and at the bottom of left panels you will see the peptide binding left, 2 alpha helices in 2 sides, and beta sheet at the bottom, like class 1. This slide shows a more realistic picture of MHC class 1 and class 2 with peptides with it. In the right panels you will see the MHC class 2, and in the left panel you will see MHC class 1 plus peptides. MHC molecules bind a variety of peptides. MHC molecules have peptide binding sites, as you noticed in previous slides, that are capable of binding peptides of many different amino acid sequences. These degenerate binding specificity contrast with the specificity of an immunoglobulin or a T-cell receptor for a single epitope. The peptide binding site is a deep groove on the surface of the MHC molecule, within which a single peptide is held tightly but by non-covalent bonds. There are constraints on the length and amino acid sequence of peptides that are bound by MHC molecules. These are dictated by the structure of the peptide binding groove, which differs between MHC class 1 and MHC class 2 molecules. The length of the peptides bound by MHC class 1 molecules is limited because the two ends of the peptide are grasped by pockets situated at the ends of the peptide binding groove. The vast majority of peptides that bind MHC class 1 molecules are 8, 9 or 10 amino acids in length. Most are 9 amino acids. The differences in peptide length are accommodated by a slight kinking of the extended conformation of the bound peptides. Features that are common to all peptides, the amino terminus, the carboxy terminus, and the peptide backbone interact with binding pockets present in all MHC class 1 molecules, and these form the basis for all peptide MHC class 1 interactions. Most peptides bound by MHC class 1 molecules also have a hydrophobic or basic residues at the carboxyl terminus, which corresponds to a complementary pocket present in the binding groove of MHC class 1 molecule. In MHC class 2 molecules, the two ends of the peptide are not pinned down into pockets at each end of the peptide binding groove. As a consequence, they can extend at each end of the groove, and so peptides bound by MHC class 2 molecules are both longer and more variable in length than peptides bound by MHC class 1. Peptides that bind to MHC class 2 molecules are usually 13 to 25 amino acids in length, and some are even much longer. Two classes of T cells are specialized to respond to intracellular and extracellular sources of infection, CD4 positive T cells and CD8 positive T cells. Circulating alpha beta T cells fall into one of two mutually exclusive classes. One is defined by expression of the CD4 glycoprotein on the cell surface, and the other by expression of the CD8 glycoprotein, as you can see in this picture, and they compare CD4 and CD8 structure with each other. These two classes of T cell have different functions and deal with different types of pathogen. CD8 positive T cells are cytotoxic, and their main function is to kill cells that have become infected with a virus or some other intracellular pathogen. This response prevents the multiplication of the pathogen and further infection of healthy cells. The general function of CD4 T cells is to help other cells of the immune system to respond to extracellular source of antigen or infection. Different aspects of this response are carried out by two subclasses of CD4 T cells, Th1 and Th2. The H in the names stands for helper, and CD4 T cells are often also known as helper T cells. Th2 cells are involved mainly in stimulating the B cells to make antibodies which bind to extracellular bacteria and virus particles, whereas a function of Th1 cells is to activate tissue macrophage to phagocytose and kill extracellular pathogens, and to secrete cytokines and other biologically active molecules that affect the course of the immune response. The human immunodeficiency virus HIV, as you know, causes acquired immunodeficiency syndrome, selectively infects CD4 T cells by exploiting the CD4 molecule as its receptor. On binding to CD4 on a T cell surface, the virus gains entry to the cell. Let me add this important point that MHC molecules are crucial in ensuring that the appropriate class of T cell is activated in response to a particular source of infection. These are two different classes of MHC molecule, MHC class 1 and MHC class 2, and each presents peptides from one kind of antigen source to one type of T cell. MHC class 1 molecules present antigens of intracellular origin to CD8 positive T cells, whereas MHC class 2 molecules present antigens of extracellular origin to CD4 positive T cells. The basis of this correlation is a specific molecular interactions between the CD8 glycoprotein and MHC class 1 molecules, and between CD4 and MHC class 2 molecules. These occur when the T cell receptor recognizes its specific peptide, MHC molecule, peptide plus MHC molecule, because of their close involvement in antigen recognition. The CD4 and CD8 molecules are called T cell co-receptors. The immunoglobulin-like domains of MHC class 1 and class 2 molecules are not just a support for the peptide binding site. They also provide binding sites for the CD4 and CD8 co-receptors, as you see in this slide. Thus, the sites on the MHC molecule that interact with the T cell receptor and the co-receptor are separated, allowing the simultaneous engagement of both T cell receptor and co-receptor by an MHC molecule. Regarding this very important and explanatory slide, it should be said that T-cell responses are guided in appropriate directions by differential expression of the two classes of MHC molecules in human cells. Virtually all the cells of the body express MHC class 1 molecules constitutively, and as all cell types are susceptible to infection by viral pathogens, this enables comprehensive surveillance by CD8 positive T cells. As you see, the erythrocyte is one cell type that lacks MHC class 1, property that might facilitate its persistent infection by malaria parasites. MHC class 2 molecules are, in contrast, constitutively expressed on only a few cell types, which are cells of the immune system specialized for the uptake, processing, and presentation of antigens from the extracellular environment. This distribution is consistent with MHC class 2 function, alerting CD4 T cells to the presence of extracellular infections. To be effective, this function need not be performed by every cell within a tissue or organ, but requires only a sufficient number of cells equipped to guard the extracellular territory. These professional antigen-presenting cells are dendritic cells, which are supremely specialized for antigen presentation and the activation of T cells, macrophages, which take up antigens by phagocytosis and endocytosis, and B cells, which efficiently internalize specific antigens bound to their surface immunoglobulin. During the course of an immune response, cells can increase the synthesis and cell surface expression of MHC molecules beyond constitutive levels. This upregulation, which enhances antigen presentation and T cell activation, is induced by several cytokines produced by activated immune system cells, in particular the interferons. In addition to increasing the levels of constitutively expressed MHC molecules, interferon gamma can induce the expression of MHC class 2 molecule on some cell types that do not normally produce them. In this manner, presentation of antigen to CD4 T cells by MHC class 2 molecules can be induced and increased in infected or inflamed tissues. This is a very nice slide. The peptide antigens that are bound and presented by MHC molecules are generated inside cells of the body by the breakdown of larger protein antigens. Proteins derived from intracellular and extracellular antigens are present in different intracellular compartments, which is nicely shown in this simple slide. They are processed into peptides by two intracellular pathways and bind to the two classes of MHC molecule in separate intracellular compartments. Peptides derived from the degradation of intracellular pathogens are formed in the cytosol and derived to the endoplasmic reticulum. This is where MHC class 1 molecule bind peptides, in ER. In contrast, extracellular microorganisms and proteins are taken up by cells through phagocytosis and endocytosis and are degraded in the lysosomes and other vesicles of the endocytic pathways. It is in these cellular compartments that MHC class II molecules bind peptides, and in this way the class of MHC molecule labels the peptide as being extracellular or intracellular in origin. In this slide we can distinguish between two compartments. When viruses infect human cells, they exploit the cell's ribosome to synthesize viral proteins, which are therefore present in the cytosol before being assembled into viral particles. In response, the infected cell uses its normal processes of breakdown and turnover of cellular proteins to degrade some of the viral proteins into peptides that can be bound by MHC class I molecules and presented to CD8 T cells. As I said, proteins in the cytosol are degraded by a large protein complex called the proteasome, which has several different protease activities. It consists of 28 polypeptide subunits, each of 20 to 30 kilodalton molecule weight. Once formed, the antigenic peptides are transported out of the cytosol and into the endoplasmic reticulum. Transport of peptide across the endoplasmic reticulum membrane is accomplished by a protein called the transporter associated with antigen processing, TAP, which you can see it in this slide, embedded in the membrane. TAP is a heterodimer, consists of two structurally related polypeptide chains, TAP1 and TAP2. Peptide transport by TAP is dependent on the binding and hydrolysis of ATP, properties shared with the other transporters in this family. The type of the peptide that are preferentially transported by TAP are similar to those that bind MHC class I molecules. There are 8 or more amino acids and have either hydrophobic or basic residues at the carboxy terminus. When MHC class I heavy chains first enter the endoplasmic reticulum, they bind the membrane protein known as cholinesterase, which retains the partly folded heavy chain in the endoplasmic reticulum, as you see in this figure. Cholinesterase is a calcium-dependent lectin, a carbohydrate binding protein that retains many multi-subunits glycoproteins, including T cell receptors and immunoglobulins, in the endoplasmic reticulum until they have folded correctly. When the MHC class I heavy chain has bound beta-2 microglobulin, cholinesterase is released from the alpha beta 2 microglobulin heterodimer to be replaced by a complex of proteins, one of which, called reticulin, resembles a soluble form of cholinesterase and probably has a similar chaperone function. A second component of the complex, called taposin, binds to the TAP1 subunit of the peptide transporter, positioning the partly folded heterodimer of heavy chain and beta 2 microglobulin to await receipt of a suitable peptide from the cytosol. On binding peptide, the now completely assembled MHC class 1 molecule is released from all chaperones and leaves the endoplasmic reticulum in a membrane-bound vesicle. It then makes its way through the Golgi stacks to the plasma membrane. Most of the peptides transported by TAP are not successful in binding to MHC class 1 molecule and are cleared out of the endoplasmic reticulum later. So far we discussed in detail the presentation of MHC class 1 plus intracellular antigen. What about extracellular antigens and MHC class 2? Newly synthesized MHC class 2 alpha and beta chains are translocated from the ribosome into the membrane of the endoplasmic reticulum. There an alpha chain and a beta chain associate with a third chain called the invariant chain, which you see in this picture, the first left panel. This is so-called because it is identical in all individuals. It is invariant, whereas the alpha and beta chains vary from one person to another, but the invariant chain is not so. One function of the invariant chain is to prevent the peptide binding site formed by association of an alpha and beta chain from binding peptides present in the endoplasmic reticulum. These peptides are therefore targeted to MHC class 1 molecules only. A second function of the invariant chain is to deliver MHC class 2 molecules to endocytic vesicles where they bind peptide. These vesicles, which have been called M2C for MHC class 2 compartment, contain proteases, for example cathepsin S, that selectively attack the invariant chain. A series of cleavage leaves just a small fragment of the invariant chain that covers up the MHC class 2 peptide binding site. This fragment is called class 2 associated invariant chain peptide, in abbreviation CLIP. Removal of CLIP and binding of peptide is aided by the interaction of the MHC class 2 molecule with a glycoprotein in the vesicle membrane called HLA-DM. HLA-DM resembles an MHC class 2 molecule in structure but does not bind peptides or appear on the cell surface. HLA-DM catalyzes the removal of CLIP and then allows the MHC class 2 molecule to sample other peptides until it finds one that binds strongly. Once the MHC class 2 molecules has lost its invariant chain and has bound the peptide, it is carried, as you see in the picture, to the cell surface by outward going vesicles. A few words about the genetic of HLA or MHC and the gene structure of MHC. The HLA complex consists of about 4 million base pairs of DNA on the short arm of chromosome 6, which is shown in this slide, and is divided into three regions. The class 1 region at the end of the complex farthest from the chromosome's centromere contains the 6 expressed HLA class 1 genes as well as several non-functional class 1 genes and gene fragments. At the other end of the complex is the class 2 region, which contains all the expressed class 2 genes and several non-functional class 2 genes. Separating the class 1 and class 2 regions is megabase region of DNA called the class 3 region or the central MHC. Although dense with other types of gene, it contains no class 1 or class 2 genes. Notably absent from the HLA complex is the gene encoding beta-2 microglobulin. I mentioned briefly at the beginning of the lecture the invariant light chain of HLA class 1 molecules, which is located on human chromosome 15, beta-2 microglobulin gene. For each HLA class 2 isotype, the genes encoding the alpha and beta genes are called A and B respectively. For example, HLA-DMA and HLA-DMB. When there is more than one gene, including non-functional genes, a number in series is added. For example, HLA-DQ1 and HLA-DQ-A2. A1 and A2 in HLA-DQ. The genes for the alpha and beta chains of the HLA-DN, HLA-DP, HLA-DQ, and HLA-DR class 2 isotypes clustered together in different subregions within the class 2 region of the MHC. This is the short arm of chromosome 6 with more details. This slide shows polymorphism in HLA genes. In contrast to immunoglobulins and the T-cell receptors, the MHC class 1 and class 2 molecules are encoded by conventionally stable genes that neither rearrange nor undergo any other developmental or somatic process of structural change. The inherited diversity of MHC molecules has two components. The first component is provided by gene families consisting of multiple similar genes encoding the MHC class 1 heavy chains, MHC class 2 alpha chains, and MHC class 2 beta chains. The second component is genetic polymorphism, which is the presence within the population of multiple alternative forms of a gene. The products of the different molecules in MHC class 1 or class 2 family are called isotypes. The different forms of any given gene are called alleles, and their encoded proteins are called allotypes. when considering the diversity of MHC class 1 or 2 molecules that arises from the combination of multiple genes and multiple alleles. The term isoform can be useful to denote any particular MHC protein. The numerous alleles of certain MHC class 1 and class 2 genes and the many differences that distinguish them make these MHC genes stand out from other polymorphic genes, and they are therefore said to be highly polymorphic. MHC class 1 and 2 genes that have no polymorphism are described as monomorphic, and genes having a few alleles are described as oligomorphic. An important consequence of genetic polymorphism is that an individual can inherit different forms of the gene from their two parents, in which case they are said to be heterozygous. When they inherit the same form of the gene from both parents, they are said to be homozygous. In humans, there are 6 MHC class 1 isotypes, HLA-A, B, C, E, F, and G, and 5 MHC class 2 isotypes, HLA-DM, DQ, DP, DO, and DR. Of the class 1 isotype, HLA-A, B, and C are highly polymorphic, and their function is to present antigens to CD8 T cells and to form ligands for receptors on natural killer cells. HLA-E and HLA-G are oligomorphic and form ligands for NK receptors. HLA-F appears to be monomorphic and remains intracellular. Its function is unknown. The human MHC class 2 isotype also display a range of properties. The 3 highly polymorphic molecules, HLA-DP, DQ, and DR, are those that directly present peptide antigens to CD4 cells, whereas the oligomorphic HLA-DM and HLA-DQ molecules have functions that regulate peptide loading of HLA-DP, DQ, and DR. The numbers of alleles currently known for each HLA locus are listed in this figure, which should be, of course, updated. The polymorphism of the HLA-A, B, and C molecules is the property of the heavy chain, beta-2 microglobulin being monomorphic, and it's not polymorphic. By contrast, the polymorphic HLA class 2 isotypes differ in diversity contributed by their alpha and beta chains. In HLA-DR, the alpha chain contributes almost no diversity, and the beta chain is highly polymorphic, whereas for HLA-DP and HLA-DQ class 2 molecules, both the alpha and beta chains are polymorphic. Overall, there is greater diversity in HLA class 1 molecules than in HLA class 2 molecules, as you can see in this slide. This slide clearly shows that MHC polymorphism affects the binding and presentation of peptide antigen to T cells. The alleles of highly polymorphic MHC genes encode proteins that differ by 1 to 50 amino acid substitutions. The substitutions are not randomly distributed within the sequence, but are mainly in the domain that bind peptide and interact with the T cell receptor. The alpha 1 and alpha 2 domains of MHC class 1 and the alpha 1 and beta 1 domains of MHC class 2. More specifically, the substitutions focus at positions within those domains that contact either bound peptide or the T cell receptor. Not all the contact residues vary, as is apparent from the HLA-DR molecule, in which the alpha-1 domain is invariant. It's not polymorphic. In contrast, variability occurs in both the alpha-1 and beta-1 domains of HLA-DP and HLA-DQ molecule. Thank you for your attention.